Good morning. Welcome to biology class. Today again we'll continue chapter biotechnology, principles and processes. And today we'll be going to learn the process of recombinant DNA technology. Now recombinant DNA technology, it involves several steps like isolation of genetic material or the DNA, separation of isolated DNA fragments, cutting segments of isolated DNA at specific sites by restriction enzymes, amplification of gene of interest using polymerase chain reaction, then insertion and ligation of DNA segment, insertion of recombinant DNA, selection of transformed cells, culturing transformed host cells and culturing transformed host cells to obtain desired product at large scale. Number 1. Isolation of genetic material or the DNA. Now, it is the process of obtaining genomic DNA or plasmid vector DNA in pure form from the bacterial cell or the host cells. So, it also involves several steps. So, number 1 is this isolation of DNA to break open the cell by dissolving cell wall. So, to obtain isolated DNA, we have to open the cell wall by dissolving it. So, for bacterial cells, they are treated with lysosomes. Plant cells, they are treated with enzyme cellulase. And fungus cells, they are treated with enzyme chitinase to dissolve. Now this dissolving of cell wall releases DNA with macromolecules present in the cells like RNA, proteins, polysaccharides and lipids. Now in eukaryotic cells, DNA is associated with histones. Now, second step involves the removal of macromolecules. RNA is removed by treating ribonuclease enzyme. Proteins are removed by treating protease enzyme, and other macromolecules, they are removed by different treatments. Now, third step involves the precipitation of this purified DNA from the medium, and we have to add chilled ethanol which causes precipitation of DNA. Now, after this, fine threads in the suspension are there which are of DNA, and these can be removed by spooling. Now, this isolated DNA again involves separation process. Now the fragments of DNA, they are separated by agarose gel electrophoresis method. Because DNA fragments, they are negatively charged, they move towards the positive electrode or anode under an electric field. Now the separated DNA fragments, they can be seen when they stained with fluorescent dye, ethidium bromide, and followed by exposure to ultraviolet radiation. They form orange colored bands, and these bands now cut from agarose gel and they are extracted by the process called elution. Now these purified DNA fragments, now they are used for constructing recombinant DNA, or they are amplified into numerous copies by polymerization chain reaction. So what is elution, children? Elution is the process of removing adsorbed material from adsorbent by washing it in a liquid. And the solution consisting of adsorbate dissolved in eluent, it is called elevate. The third step, cutting segments of isolated DNA at specific locations by restriction endonucleases. Now these restriction endonucleases, they are the molecular scissors which cut the DNA into smaller DNA fragments which contains one or few genes. Because endonuclease, it recognizes sequences and it occurs many times within the source DNA, cleavage will produce large number of fragments of different sizes. So what is gel electrophoresis? It is a technique for the separation of enzymes, proteins, amino acids, nucleotides and nucleic acids which are based on the differences in their net charges in an electric field. Two types of gels are used, agarose gel and polyacrylamide gel. Agarose gel is used for the separation of medium and large size nucleic acid molecules. Polyacrylamide gel, it is used for the separation of proteins and purification of enzymes or separation of DNA fragments. Now here it is an apparatus for gel electrophoresis. It is made up of glass plates. Agarose gel is placed here, and with the help of the dropper, the DNA fragments, they are placed in the particular apparatus. It is connected with a electric field, and large and small size fragments, they are placed according to the size. Longer size they are on the upper side and shorter size they are lower side. Now amplification of gene of interest using PCR or the polymerase chain reaction. Now it is the method in which multiple copies of gene of interest in vitro method is used by using two sets of primers, oligonucleotides and thermostable enzymes. DNA polymerase isolated from a bacterium Thermus aquaticus Carimulis developed PCR in 1985 and she received the Nobel Prize in 1993. Now polymerase chain reaction involves three steps: denaturation, then annealing of primers, and third is extension of primers. So denaturation or melting of target DNA. So an excess of primer is mixed with target DNA fragment of the gene which is to be amplified. It is heated at 98 degrees Celsius. Now the two strands of DNA fragments, they get dissociated to form single strand, and each single strand of DNA now act as a template for DNA synthesis. Second step involves annealing of primers. Now this solution is allowed to cool to about 60 degrees Celsius. Normally when the cooling process is going on, the separated strands of DNA reassociate into double-stranded DNA. But here, the excess of primer, some separated strand base pair with their complementary primers. The third step involves extension of this primer. Thermostable DNA polymerase enzyme is added in the solution along with 4 types of nucleoside triphosphate. Now using this nucleotides and genomic DNA as a template, the enzyme DNA polymerase, it extends the primers to make a complementary strand on each separated strand of genomic DNA, as in DNA replication. Now, if this process of vitro DNA or artificial DNA replication is repeated many times, the genomic DNA can be amplified to 1 billion times within no time. So here the polymerase chain reaction. First we select the region which is to be amplified. Okay. Now then we heat the particular DNA strand, or we can say we do the denaturation process. So after heating, it breaks up into 2 different strands with the primers. Now again this primers has to be extended. Okay. So after extending primers, this amplified DNA is obtained. So this is all for today, children. Rest of the steps we'll learn in our next class. Till then, thank you and have a nice day.